Hello, my name is Alexis, and this is the phospholipid bilayer. Here we have the extracellular space. The intracellular space is on this side, and attached to the phospholipid bilayer in between it was the G protein coupled receptor, also known as the GPCR. GPCRs interact with G proteins in the plasma membrane. The signaling cascades of a GPCR initiate from the binding of an external signaling molecule in the form of a ligand or other signal mediator. This causes a conformational change in the receptor and triggers the interaction between GPCR and a nearby G protein, leading to the activation of the overall G protein. This process is regulated by guanine nucleotide binding complex consisting of the alpha G protein, the gamma G protein, and the beta G protein, in addition to other regulatory proteins. In the inactive state, the GPCR is bound to a heterotrimeric G protein complex, otherwise already stated here. The alpha G subunit is bound to GDP and is firmly attached to the beta G protein and the gamma G protein, as you can see there. Binding of a ligand to GPCR results in the conformational change of the receptor, which leads to the transmission of the alpha G protein, to active status, along with the exchange of GDP to GTP, as well as the dissociation of the alpha G protein subunit to the beta and gamma protein subunits and from other receptors. The signal is then terminated by the hydrolysis reaction of GTP, therefore restoring another heterotrimeric G protein to form new G protein binding complex, which is not otherwise drawn on here, but it just leads on to another signal transduction in another part, and then the cascade continues to lead on to another one and another one, leading to more signal transductions. The dissociated alpha G protein and the beta and gamma G protein subunits will interact with other intracellular proteins to continue cascades of signal transduction.